The cardiovascular system, this particular lecture, starts section 4. So this is lecture 4-1, and we are going to introduce you to the cardiovascular system, which includes the heart, the blood vessels, and blood. And we are going to start off really with blood, its constituents or its makeup. So the body has to be in constant communication with the internal and external environment. All parts of the body are linked by the cardiovascular system. The cardiovascular system is basically the transportation system for substances that we need from that external environment. And sometimes we need to take substances from our internal environment and eliminate them through waste, and the cardiovascular system plays a role in that. Also, whenever substances are made by our cells, they need to be delivered to other parts of the body or to other organs, and it is the cardiovascular system that is able to accomplish that. Now, when you think about it, embryos, I do want to mention that embryos do not need a cardiovascular system. Because they're so small that everything gets by through diffusion, or maybe I should say that substances are transported through diffusion. So the cardiovascular system develops a few weeks into development because at that point, as the fetus grows, then it becomes too big for diffusion. So around 3 weeks, the heart beats and blood starts circulating so that it can act as a transportation system. The function of the blood is manifold. First off, we have the fact that it transports dissolved gases, nutrients, hormones, and metabolic waste. So the blood carries oxygen from the lungs to your tissues, and it also takes carbon dioxide from the tissue to the lungs so that it can be expelled. It distributes nutrients, carries hormones from the endocrine glands to target cells, and takes away waste to kidneys for your excretion. So it's, as you can see, the blood is very, very dynamic in what its function is. Blood is able to absorb and neutralize acids like lactic acid, which is developed during fermentation. And it also regulates excesses and deficiencies of ions. So it is able to regulate the pH and the ion makeup of fluids. Blood restricts fluid loss at the injury sites through clotting. So clotting basically works as a temporary patch, and later on in the lecture, we will cover how blood clots form. And blood also defends against toxins and pathogens. It's able to transport white blood cells, which fight infection, to remove debris. And blood absorbs heat, so it can stabilize body temperature. It does so by absorbing heat that's made by skeletal muscle and redistributes it to other tissue. If you have a high body temperature, the blood goes to the skin surface to release heat. And if there's a low body temperature, flow of warm blood is restricted to crucial structures like your brain and other temperature-sensitive organs. Since we've already done the blood lab, and because we have covered histology earlier during this class, earlier in the semester, hopefully you would remember that blood is a connective tissue. It's a fluid or a liquid connective tissue. And it has plasma. It consists of plasma and also formed elements. And the formed elements are red blood cells, which I'll label as RBCs, white blood cells, and platelets. No matter the source of blood, wherever the blood is coming from, blood basically has the same physical characteristics, regardless of its source. Its temperature is a little above body temperature, which is 38 degrees Celsius. And this shows looks like a O here, but it should be a superscript as 38 degrees Celsius. And that's a little above body temperature. By the way, that 38 degrees Celsius it translates into about 100.4 degrees Fahrenheit, just so that you can have a reference. Its viscosity, it's viscous, so it's about 5 times stickier than water. It's way more cohesive and resistant to flow, and the stickiness comes from the interactions of proteins, elements, and water molecules in the plasma. And it has a slightly alkaline pH. We will call the average here 7.4, but there's a very limited range between 7.35 and 7.45. And if it deviates from that range, that can cause some serious problems. I just quickly, before we go into detail about the formed elements, I just want to recap here that red blood cells are one of these formed elements, and these are made in the marrow. And you also have white blood cells, which help fight disease. And of course, by the way, the red blood cell's job is to transport oxygen and carbon dioxide. So I'll just say transport gases. And then you have these cellular fragments that are called platelets. So these are cellular fragments, and you'll see that these are responsible for clotting. The red blood cells make up about 45% of your blood sample. And the platelets account for about 1% of your blood sample. And as well, so does your white blood cells. So white blood cells and platelets account for about 1%. And the plasma that's in the blood accounts for about 55%. So remember, the formed elements are red blood cells, white blood cells, and platelets, but the blood is composed of these formed elements as well as plasma. Plasma consists of about 55% of your whole blood. Together, the plasma and the formed elements are considered whole blood. If you go to the doctor for a checkup, or if you are in the emergency room or the urgent care, one of the first things that will be done is blood collection, because your blood is able to give you a general sense, or give the healthcare workers a general sense of your health. So blood is collected, typically your whole blood is collected through a vein. So that would be considered, the name for that would be venipuncture, and it's in front of the elbow usually. The common site would typically be the median cubital vein, and it's used because it's superficial, so it's easy to find, and there's low pressure there, so it heals easily after it's been punctured, because it has thinner walls than arteries, so it's easy to puncture, fast to heal, easy to find. Sometimes blood could be collected from a fingertip or earlobe, so we call these peripheral capillaries. And in the blood lab, we collect it from the fingertip, and sometimes it's collected from an arterial puncture. And this is done when we want to evaluate gas exchange efficiency in the lungs. Okay, so now remember that we are talking about the composition of blood. Blood is a fluid connective tissue, and it's composed of plasma as well as the formed elements. Right now we're focused on the plasma, and the plasma contains plasma proteins. There are 3 main types, and these plasma proteins are the most abundant of the dissolved substances that are inside of the plasma. And these proteins remain in the blood and the interstitial fluids And ordinarily, they are not used as energy sources. The three main types of plasma proteins are albumins, fibrinogens, and we also have the globulins. In a few more slides, we'll be discussing the specifics of these plasma proteins. Within the plasma, there are also hormones, and there's also nutrients like amino acids and nucleotides, lipids. We also have gases like oxygen and carbon dioxide, and of course, water. The most important blood gases, because there are others, but the most important blood gases are oxygen and carbon dioxide. And plasma also contains a considerable amount of dissolved nitrogen, which ordinarily has no physiological function. The plasma, once again, I'll just back up here to nutrients. Okay, the plasma has nutrients, which includes amino acids. I'll just write that as an AA. You have amino acids. You also have some sugars, simple ones. You might have, you not might, but you have nucleotides and lipids. Nucleotides are the building blocks of nucleic acids. And you also have some lipids there. These are all absorbed from the digestive tract. And so, just to give you an example of how gases and nutrients might be used or transported in the blood, you might have plasma that would transport glucose from the small intestine to the liver so that it can be stored as glycogen or converted to fat. If blood glucose concentrations drop below the normal range, then glycogen might be broken down into glucose. So you can see that there's some serious communication going on and some serious transport happening to make sure that all of the cells are able to maintain homeostasis. Okay, so now we're going back to the dissolved substances which are in the plasma, and the most abundant one would be the plasma proteins. These are the most abundant plasma proteins are. Okay, they're more abundant than nutrients and gases and all of that inside of the plasma. So there are three types of plasma proteins, three main types, and that would be, we'll start off with the albumins. Now, the albumins are the smallest in size of the proteins, but they make up, they're the most abundant when we talk about weight. So they're the smallest in size, but they are the most abundant, which is important to remember. Okay, they make up about 60% of the proteins by weight. As far as the weight, they make up about 60% of the weight. These are made in the liver, and they are they're very plentiful, and albumins are an important determinant of the osmotic pressure of the plasma, and that's because they are so plentiful. Okay, so just a little note on this. The hopefully you can remember how water moves as far as from a high to a low concentration. So water will always move to greater osmotic pressure. If the concentration of these albumins fall, then the osmotic pressure will also fall. And if the osmotic pressure will fall in the plasma, and if the osmotic pressure falls there, that means that there will be greater pressure in the tissues, in your tissues, and so that means that water will move to your tissues. So if the concentration of the plasma proteins fall, the tissues would swell, and that condition is known as edema. So it's very important the role that albumins play in maintaining osmotic pressure. Edema can result from starvation or a protein deficient diet or from an impaired liver that cannot synthesize the plasma proteins. The other type of protein would be the globulins, which make up about 36%. And these act as transport proteins. They can be further subdivided into alpha, beta, and gamma globulins, but for our class, I don't want you to have to worry about that. You don't have to remember those. The liver synthesizes these globulins, and there are a variety of functions, including transport of lipids and fat-soluble vitamins. And they also transport antibodies. Lastly, we have fibrinogens, and fibrinogens make up a very small amount. They only make up about 4%. And the fibrinogens you're going to see later when we talk about clotting, because that's what they function. They function in blood coagulation. What happens is fibrinogen converts to fibrin, and fibrin forms the basic framework of a blood clot. As we go along, or as you read, you might sometimes hear the term serum. And serum is simply the plasma, so the fluid, that liquid, but the plasma without the clotting proteins like the fibrinogen. So that's what serum is, and 90% of the plasma, as you would have heard me mention, you heard me mention that they are synthesized by the liver. And sometimes liver disorders can result in the altered composition of blood and altered function of blood. And that's because if the liver malfunctions, well, you have a whole bunch of plasma proteins that are not going to be made properly and won't be able to do their job. So we just talked about the plasma, and we know that that is one part of this connective tissue, but the other part will be formed elements. And one of the formed elements would be the red blood cells. The red blood cells make up the largest part of all of the blood cells. It actually makes up 99%. And I was about to write it there for you, but realized I didn't have to because I already have it in there. So it makes up about 99% of the formed elements. The red blood cells are also called erythrocytes. And the hormone erythropoietin is what is responsible for controlling the rate of red blood cell formation. As a matter of fact, if there is like a level drop in the oxygen, if there's less oxygen, the more hormones, the more erythropoietin that will be released in the kidneys and liver, because the more erythropoietin, then the more red blood cell production, which means that there can be a more efficient pickup of oxygen. A blood sample is usually about 45% red blood cells by volume, and that percentage is called the hematocrit. And the hematocrit in men and women are slightly different. In men, it's about 46%, but in women, it's about 42%. And overall, the red blood cell count in men are 5.4 million versus women that have 4.8 million per microliter. Of course, there should be a range given for that, but this is about the average. But healthy people do vary in their range. Red blood cells contain a molecule called hemoglobin, and the hemoglobin is what's responsible for transporting oxygen and carbon dioxide. And in a few slides, we will get into the hemoglobin structure and function, which is actually quite fascinating. The red blood cell has this sort of biconcave shape, which I attempted to draw so very horrible. If you look at this portion that I'm about to trace in reddish color, you'll see that it has basically the red blood cell, this biconcave shape, is an adaptation for transporting gases. It increases the surface area, And also, it gives the cell a particular flexibility to squeeze through narrow capillaries. Also, this particular shape places the cell membrane, which I would have on the cross section of the cell, I just drew it in red, but it also places the cell membrane right here closer to that oxygen carrying molecule that I introduced you to earlier, called hemoglobin. So this indentation puts the plasma membrane closer to that molecule, which you'll see is very important. The hemoglobin access is extremely important. Now, red blood cells, they are unable to go through cell division because as they are formed, once they mature, some organelles are lost, including the nucleus. So that nucleus is rather large, so the red blood cell, once it's mature, it ejects it so that efficient gas exchange can occur and there won't be a whole bunch of organelles taking up space there. So because it loses many organelles, because the organelles are lost, the red blood cell relies on glucose from the plasma to get its energy. Now, this is a really wonderful, very outstanding adaptation, the fact that it relies on the plasma for energy. And this is because during respiration, the most amount of energy is made in the very last step where oxygen is used. Can you imagine if this red blood cell had to do cellular respiration and use oxygen? Then that means it would be using the oxygen that it was designed to take and transport to other tissues. So it's wonderful that it does not use oxygen. It's wonderful that it does not do cellular respiration. It does not have the mitochondria to do cellular respiration, because then it will be using all the oxygen that it picked up to go take somewhere else. So this figure has diagrammed the picture of the red blood cell, and this, of course, is not as great of a picture as the one I drew before. Okay, so I guess that's arguable, but it shows you this biconcave shape and the fact that the red blood cell is three-dimensional. And again, this is a wonderful adaptation so that the membrane is placed close to the hemoglobin that would be in this area that I just circled in, or that I just drew in rather. And of course, the shape allows the blood cells to squeeze past areas that are quite a tight fit. Okay, so now let's move on to hemoglobin. Each blood cell is about a third hemoglobin. Each red blood cell is about a third hemoglobin by volume. And this is what makes the blood red. Red blood cells are filled with this hemoglobin and is like an oxygen and carbon dioxide shuttle bus. So hemoglobin transports oxygen around the body, and it also picks up carbon dioxide and takes it to the lungs so that it can be removed. Molecules of hemoglobin account for about 95% of of a red blood cell's intracellular proteins. Again, it's a transport mechanism. It picks up oxygen in the lungs and carries it to the tissues, and it also takes carbon dioxide from the tissues, picks that up, and take it to the lungs so that it can be dispelled. The hemoglobin bus releases oxygen passengers to organs. So that's what I like to think of. I like to think of the hemoglobin as a big bus, which let's just kind of draw a big, you guys know how wonderfully I draw. Okay, so that's your bus. And this is actually hemoglobin. And I'm just going to write HB. So it's our hemoglobin bus. The hemoglobin bus has these passengers in there. So here's the passengers. Oh, I just switched colors by accident. Here's your passengers. Oh, but goodness. Sitting down in the bus. Okay, so these would be oxygen. And they also have carbon dioxide passengers. But let me tell you about the seats that they sit in, okay? So each molecule of hemoglobin has iron in it. Okay, so I'm going to draw the iron seats. Here they are in blue, right? So essentially, each molecule of hemoglobin has a four polypeptide chain, we call it, okay? So it's protein, and it has four molecules of iron in it. So there are really four iron chairs in here, iron seats in there. And the iron acts like seats so that the oxygen could sit in it. Here's more oxygen passengers, oxygen passengers sitting there. Maybe we should draw some hands on there. Man, this drawing is amazing. There we go. Okay. And this hemoglobin bus is going to go to the organs and to tissue that need the oxygen and drop those passengers off. Then, when it's empty, that hemoglobin bus will return to the lungs and load up on oxygen again. So I'm hoping that you see how very important the iron is here, because the iron will be the seats, okay? And if you are someone who is iron deficient, you have low iron, then that means that you don't have enough seats for the oxygen to sit into, which means that less oxygen would be transported. Something else I want to point out is that the hemoglobin structure also contains heme, okay, and it's the heme that would bind to oxygen, but carbon dioxide also has to be transported. So those are bound to what we call globular subunits, which also makes up hemoglobin. Now, this is really interesting because that way carbon dioxide and the oxygen do not have to compete to sit in the same seats. So the heme binds oxygen, and remember the heme has that iron atom. The iron is where the oxygen sits into. And then the carbon dioxide binds to the globular subunit. So those will be some other seats in here that I'm drawing in in green. And then that way, oxygen and carbon dioxide do not have to compete. When you need oxygen, you want oxygen. You don't want carbon dioxide sitting in those seats. When oxygen is bound to hemoglobin, we call it oxyhemoglobin. And oxyhemoglobin is bright red. So you have oxyhemoglobin once that bond takes place, and that will be a bright red color. So oxygenated blood is bright red. But then when the oxygen is released, then that would result in something called deoxyhemoglobin. And deoxyhemoglobin would be a much darker color. Almost gives it sort of a bluish tinge. Now that oxygen-heme bond is extremely weak, and that's great because hemoglobin does not hold on to oxygen so tightly that it is not going to let it go. When hemoglobin encounters an area with a high concentration of oxygen, like in the lungs, all of the compartments bind to the oxygen molecules. So all of the iron seats pick up oxygen. When the hemoglobin goes to an area where there's not a large supply of oxygen, especially when you exert yourself, the hemoglobin in the vicinity releases the oxygen, which is soaked up by that deprived tissue, and it's going to be soaked up to make ATP. So once again, when the hemoglobin encounters an area where there is high oxygen, it's going to get loaded up with oxygen, and as it goes around or as it travels throughout the body, when that hemoglobin comes into contact with low oxygen and high CO2, the hemoglobin is going to release that oxygen that it picked up previously. And it also picks up that high carbon dioxide, picks up the carbon dioxide and takes it back to the lungs. Sometimes an individual may have a reduced ability to carry oxygen, a reduced capacity to carry oxygen. And this can happen because of a low hematocrit or low hemoglobin. Remember that in the lab, when you guys did the blood lab, you guys measured the hematocrit and the hemoglobin, and both of these tests told us how many red blood cells were present. So if the blood has a low hematocrit or a low hemoglobin, then that means that the hemoglobin is reduced as far as its content. The amount of hemoglobin that's there is reduced. Then the oxygen carrying capacity of the blood will be reduced also. And when this happens, we call it anemia. We say that a patient is anemic. The red blood cell is exposed to a lot of stress and friction. I mean, when you think about it, it's going to be moving and squeezing through capillaries. It's bouncing against walls of the blood vessels. And all of this stress and friction that it goes through. And the fact that the red blood cells, when you think about it, is taking one round trip from the heart through the tissue and back to the heart in less than 1 minute. That is a very fast trip. And so the red blood cells are being forced and bounced and colliding with other vessels. And there are no repair mechanisms. So red blood cells only have a lifespan about 120 days. And on any given day, about 1% of your red blood cells are being replaced. And new red blood cells come into circulation at about the same rate. When red blood cells age or when they get damaged, some rupture or hemolyze, and they will break down. The hemoglobin breaks down in the blood. And the subunits that we mentioned earlier break down and are carried away by your urine. If this happens at a high rate, urine becomes really reddish. Fortunately, only about 10% of the red blood cells survive long enough to rupture or hemolyze, and when that happens, they are engulfed and most of the components are recycled. Next, we're going to talk about the 3 steps to recycling the hemoglobin. Okay, so once the red blood cells are engulfed, or if a red blood cell is engulfed, each component of the hemoglobin molecule has a different fate. So, of course, you would remember that the hemoglobin molecule is made up of globular proteins. Those globular proteins are broken down into amino acids, and then, of course, the amino acids, those are the building blocks for other proteins as well. So those are simply used later on in some other process in building protein. Then there is the heme. The heme is stripped of its iron, and the heme is converted to biliverdin, and the biliverdin is converted into bilirubin, which is then sent over to the liver. It's absorbed it's released into the bloodstream, and then the liver cells absorb it and release it into the small intestine into bile. So if your bile ducts are blocked by gallstones, for example, then the bilirubin diffuses into the tissue, and that's what gives you a yellow appearance, and we call it jaundice. The bilirubin is just to make sure you know what it is. It's just a simple waste product, and it is a product once the red blood cells have broken down. And that's just going to pass, like I said, through the liver, eventually out of the body, mostly in feces, a small amount in urine as well. Then there is that iron that got stripped off the heme. The iron is taken from the heme molecules, and it's either stored in macrophages or released into the bloodstream. And if that happens, it binds to transferrin. And the transferrin, these are used to make new hemoglobin molecules. Transferrin is a plasma transport protein, and it is used to make new hemoglobin molecules. So these are the fates of all of the components of the hemoglobin molecule once it is broken down, engulfed and broken down. So this picture is diagramming what happens to each of the components that we just discussed. Here you have the old damaged red blood cell, so it ruptures. And what happens, of course, is that red blood cell, you'll have the amino acids that it breaks apart into, and the amino acids are, they're broken into amino acids, and those are used to make some other proteins. And then you have the heme molecule right here, which is stripped of its iron, and that iron is taken from the heme and stored in either a macrophage. This purple is a macrophage in the liver, or it is released into the bloodstream. So if it's released into the bloodstream, it binds to transferrin right here, and then that transferrin is used to make new hemoglobin proteins. So here we have the formation of new red blood cells. Here's a new red blood cell, and that's going to be put into circulation. Next, you have the heme molecule. And this heme molecule was left after the iron was stripped from it. And so that is going to be converted into biliverdin, which is converted into bilirubin. And then that's released into the bloodstream. And the liver cells absorb it and release it into the small intestine. So here's the liver cells, and then it's excreted in bile. And most of it is eliminated in feces as well as urine. As far as the iron that we find in our blood, which is again, it's attached to our heme, the men have about 3.5 grams of iron, and 2.5 of the grams is found in hemoglobin. So that means that there's about a 1 gram reserve for men. And women have smaller iron reserves than men, only about half of a typical male. So here we have a reserve of only 0.5. Why? Because we have about 2.4 grams of iron, about 1.9 is in the hemoglobin, which means you're only left with about 0.5 gram reserve. This is why a lot of times women often need dietary supplements. A lot of times you'll find that women are anemic, because remember, if you have low iron, then you don't have any seats for the oxygen to sit in. For those individuals who do become anemic, a lot of times these individuals look pale, they lack energy as well. A pregnant woman, a lot of times, can become anemic if she doesn't eat iron-rich foods, because her blood volume increases due to fluid retention to accommodate the requirements of her growing fetus. And the increased blood volume decreases the hematocrit, and that can lead to anemia. Now, there's one disorder that is and is inherited, an inherited disorder that's in contrast to anemia. This is called hemochromatosis. In hemochromatosis, instead of having too little iron, what happens is that the small intestine absorbs iron at probably like 10 times the normal rate. And the iron will just build up in the organs, and it gets to toxic levels. And so what these individuals have to do is have blood removals as often as every week following their diagnosis. And unfortunately, the blood is just discarded because it can't be used because it has too much iron in it, so it's really not good for anyone else to have that blood. Okay, so now let's talk about red blood cell formation. And the term that's given to that is erythropoiesis, and this is red blood cell formation. There are specific names for key stages of red blood cell formation. Red blood cells come from hemocytoblasts in the red bone marrow. So hemocytoblasts are the embryonic stem cells that can differentiate into other types of stem cells. Erythropoiesis occurs in the red bone marrow, or what we call myeloid tissue, and the hemocytoblasts, they produce the myeloid stem cells, and some of these myeloid stem cells go on to mature. Erythroblasts, these are immature red blood cells that actively make hemoglobin. So these are going to be synthesized in hemoglobin. And when the nucleus is shed, they become reticulocytes. This happens about 4 days of maturing. After about 4 days of maturing, the reticulocytes then enter the bloodstream, and then they complete maturation after about 24 hours. Okay, so let's backtrack, or say once again, retrack what we just said. You have hemocytoblasts. The hemocytoblasts are in the red bone marrow, which is indicated here. And the hemocytoblasts produce myeloid stem cells, and some of the myeloid stem cells go on to mature. You have these myeloid stem cells, whereas some of them are destined to become the red blood cells eventually. So that's following the path that I just put an arrow onto here. And because I want to point out that not all hemocytoblasts will become myeloid stem cells. Some of them become lymphoid stem cells, which we will talk about later on. And then of the hemocytoblasts that become myeloid stem cells, not all of them are destined to be red blood cells. Some of them will become platelets, and you have some that will become white blood cells as well. Okay, so right now we're looking at those that will become red blood cells, and so you can see that those, you would see that they become erythroblasts, and the erythroblasts are immature red blood cells, and these are going to actively make the hemoglobin. After about 4 days, these maturing cells will eject the nucleus, as you can see done right here, circled in green. It's ejecting the nucleus because, remember, the role of the red blood cell is to be able to do oxygen and carbon dioxide transport. So these gases are going to have to be able to diffuse across the cell's membrane easily. And if the nucleus is there and other organelles, it just really takes up too much space, and we cannot do that efficiently. So it ejects it, and then what enters the bloodstream is the reticulocyte. And once that enters, it would take about 24 hours for it to complete maturation. And when it does, you have the erythrocyte. Okay, now that you know how red blood cells are made, let's move on to blood typing. In the very earlies, blood transfers had really mixed results. Sometimes a patient would improve, or sometimes they would get worse and even die. And so it was finally figured out that blood was of differing types. And of course, this is genetically determined. Blood typing allows us to classify blood based on glycoproteins that are present on the outer surface of the red blood cell's plasma membrane. These proteins are called antigens or agglutinogens. And these can trigger an immune response, and they are found on the outside of the plasma membrane, on the surface, I should say, of the plasma membrane. Agglutination, the term agglutination is clumping. That means clumping. So in this case, we're talking about the clumping of red blood cells anytime you hear the term agglutination. And a mismatched blood transfusion produces agglutination. The antigens on your cell surface, those are considered normal. As a matter of fact, during your development, your immune system would have done an inventory of what was self and what was not, and your antigens on your blood cells would have been considered normal. And just to make sure that you understand, antigen comes from the term antibody. Anti comes from antibody, and the gen comes from generator. So this will, of course, anything that generates antibodies are things that are soliciting an immune response. So your surface antigens are absolutely normal to you. The inventory was taken, they're normal, they're not foreign, so they wouldn't trigger an immune response. The problem comes when, if another type of blood is introduced into you, then that will trigger an immune response. The presence or absence of these antigens on the membrane of the red blood cell is what determines the blood type. There are three major antigens that are associated with blood. You have antigen A, B, and then there's the RH, that type, or we call it D. But the RH factor is what is being considered or referred to whenever we say negative or positive for your blood. That's the RH factor, whether you have it or not. If you have the RH antigen, then you're considered positive, and if you do not have it, you are considered negative. Okay, so if you were type A blood, and we did this lab in class already, and you guys found out your blood types. So if you are type A blood, then that means that your cells, your cell membrane, if this is your cell and you're A, that means that your antigen that's connected to your receptor is of the A type. Then you have type B blood, and type B blood would have only B antigens on the surface of the membrane. If you are type A B blood, that means that your cell had both A and B antigens on the cell's membrane. If you are type O, that means that you do not have A or B connected to your cell's membrane. Rh positive means that you have the Rh antigen, and if you are Rh negative, then that means that you do not have the Rh antigen. This is just an informational slide for you to look at the distribution of blood types of selected populations, and perhaps you could even compare yours to this. In our lab, I found out that I was O positive. So here I am. The U.S. average, of course, leans towards O, but African American, O positive. So it looks like I'm in the majority there because 49% of African Americans would be O positive. Remember, this is genetically determined. And so, or at least I just said O positive. I should have said that 49% of African Americans have type O blood. So that means we are lacking the AB antigen. Now I have Rh positive blood, so I'm O positive, and you can see here that 95% of African Americans are positive for the Rh antigen. So I fall directly where, just looking at this table, where you'd expect me to fall in the vast majority. Where do you fall? Go ahead and look it through and see where you fall as far as the percentage of your blood type. Now, inside of your plasma, you have these proteins that are called agglutinins, and another name for it would be antibodies. Okay, so you have antibodies, but these are agglutinins, and these are found in your plasma, and your agglutinins or antibodies that are in your plasma will not attack your own antigens on your red blood cells, but they will attack the foreign antigens of a different type. So if you are type A blood, then you'll contain anything that's anti-B, right? Anti-B antibodies is what your plasma will contain. If you are B blood, of course you won't have anti-B because you're B blood, but you will have anti-A antibodies. So if A blood comes into a person who has the B type, then the anti-A antibodies will grab a hold of it and clump it up. Type AB blood is not going to have any antibodies against A or B. So they do not have any antibodies in the plasma against the A or B, which is good because if you're type AB, you do not want to have antibodies that are going to attack your blood cells. So there are no antibodies in that case. And then if you have type O blood, the type O blood contains both antibodies, anti-A antibodies and anti-B antibodies, which is fine because type O has no antigens on them. So how do these cross reactions and transfusions take place? Well, anytime the antibodies in a recipient reacts with the antigen on a donor's red blood cell, then you're going to get agglutination or clumping of the red blood cells. And this is what we refer to as a cross reaction or a transfusion reaction. This is why it's very important to check the blood type before transfusion to make sure that there is compatibility so that the antibodies in the recipient will not go after the cells of the donor that are coming in. Now, I don't want you to mix up the ABO blood typing gene and the RH gene. These are both genetic. The anti-A or anti-B antibodies, these just spontaneously develop during the first 6 months of life, and no exposure to any foreign antigen is needed. It is genetic. There's no exposure needed. It is spontaneously going to develop. But the anti-RH antibodies that are in a negative person, somebody who has who tests negative for the RH antigen, they don't have to develop any antibodies against the RH antigen because they don't have the RH antigen. They are negative. But so they don't develop the anti-RH antibodies unless a negative person is exposed to RH positive blood. So exposure can occur accidentally during a transfer, but it can also happen during childbirth. So here I go with my beautiful drawings again. But let's say you have a mom who is RH negative, but she is carrying a child who is RH positive. Okay, so that's the baby in the belly. And here the mom is. Okay, beautiful drawing. She's happy because she's pregnant, getting ready to have a baby. All right, so she's RH negative. And such a pregnancy, this could be quite uneventful. However, at birth, or if there is a miscarriage, the placental membranes that separate the maternal blood from the fetal blood during the pregnancy, those can tear. And some of the infant's RH positive blood cells can enter the maternal circulation. So you can have this RH positive entering the maternal circulation, like I just illustrated here. And those RH positive cells might stimulate the maternal tissues to begin producing anti-RH antibodies. So, of course, previously this was not needed, but now, since there's exposure, the anti-RH antibodies may develop. If a woman who has already developed anti-RH antibodies becomes pregnant with a second RH positive fetus, so you have this very same setup, but now it's a second child, which I'm going to put in green, RH positive, what can happen is that these antibodies can cross the placental membrane from the mom. The mom now has our anti-RH antibodies. If it's anti-RH, that won't be good for a RH positive child, and it could then destroy the fetal red blood cells. The fetus then develops a condition that we call erythroblastosis fetalis, or a hemolytic disease of the fetus and newborn. And even though this is really rare, today it can be very devastating for a child. It's rare today because obstetricians can look at the RH status of a woman, an RH negative woman who might carry a RH positive fetus is given an injection of a particular drug, and the drug is composed of anti-RH antibodies, which bind and shield any Rh-positive fetal cells that might contact the woman's cells and sensitize her immune system. It's amazing, but it works. Okay, so if you are a type A individual, let's just delve a little deeper into this. If you are a type A blood individual, that means that your red blood cells have this A antigen on them. Okay, and so in this picture, it's illustrated as little circles. So if you have type A blood, then look at your antibodies. They're going to be anti-B. And what would happen is if this type B blood that we look here, you can see that their surface antigens are B's. If this B blood came into this individual, these antigens will immediately, the antigens of the type B blood will immediately have these antibodies grab a hold of them. As you can see, they fit. Look at it. You see the little receptor that looks like a triangle here? That can fit around that surface antigen very nicely, and it would clump it up. And or the correct term would be it would have some agglutination take place. So it would clump up, and that can hurt the recipient for sure. Here you can see that type AB has both of the surface antigens present on them, both the A's and the B's, and then type O has absolutely nothing on them. They lack those. If you're looking at this though, a question might be something like, okay, if you look at these blood groups and you look at the antibodies present, which group can receive blood from all groups? Well, that would be type AB. So we call them the universal recipients. These can receive from all groups. Of course, the best thing is always to match, make sure you have the compatible blood types. But the AB can receive from all groups because they have no antibodies. The space is clear, okay? So no anti-A antibodies, no anti-B antibodies, so nothing is going to grab a hold of this. And then type O blood, they don't even have any antigens on them, so the type AB can receive from all. Now, who can give blood to any other type? Hopefully you answered O. Type O can be considered the universal donor to all groups, and that's because they lack antigens A and B. So they will not elicit any sort of immune response from type A blood, type B blood, or type AB. And that's because they have no antigens on them. Now, the type O blood, they have both the anti-A antibodies and anti-B antibodies. So who can they receive blood from? They cannot receive blood from any other type except for O. They will reject anything with an A, and they will reject anything with a B. So that means type A, type B, and type AB cannot be given to type O. If incompatible blood was given to a recipient, the surface antigens on the donor blood will react with the antibodies in the recipient's blood, and the antibodies, they will then form around the antigens and clump up that blood, and then that can lead to hemolysis of the affected red blood cells. Okay, now we just finished talking about the red blood cells, or the erythrocytes, and so now we are going to move on to the leukocytes, also called white blood cells, and these protect against disease. Now, the white blood cells, they are larger, I should say, than red blood cells, and they're involved in immune responses, so that immune responses is protecting against disease. They do have a nucleus and other organelles, And they lack hemoglobin. The red blood cells have a hemoglobin and no nucleus, but the white blood cells have a nucleus but no hemoglobin. There are 5 types of white blood cells that are found in the blood. You have they differ in size, shape of the nucleus, and they differ in the nature of the cytoplasm. You have what we call the granulocytes, and the granulocytes are all of the phils. So neutrophils, eosinophils, basophils, those are granular cytes. These granular leukocytes, they have granular cytoplasm. Okay, and again, they're all of the phils. The agranulocytes, those have no granules, kind of looks like debris. They have no granules in the cytoplasm. So the granulocytes have granules in the cytoplasm, and the agranulocytes have no granules. A means without, so no granules in the cytoplasm. And the agranulocytes are the cytes, the lymphocytes and the monocytes. Now, the white blood cells, these are formed from stem cells, hematopoietic stem cells, and they are also in the bone marrow. On the earlier picture, when we were talking about erythropoiesis, I pointed out that some of those stem cells will become white blood cells as well. So this is all taking place in the bone marrow in response to hormones. And the blood transports white blood cells to the sites of infection. Then they'll leave the bloodstream, enter the tissue space around or outside of the blood vessels. So as I just stated, the blood, the white blood cells, they are going to be moving through the blood, but they're able to come outside of the bloodstream and go into tissue space outside of the blood vessels. And this is called diapedesis because they're going to be squeezing through, migrating out of those blood vessels into the tissue space. The white blood cells are attracted to specific chemical stimuli, and we call this chemotaxis. Specifically, we call it positive chemotaxis because they're going to be moving towards the chemicals, and this is what guides them to pathogens. Neutrophils, eosinophils, and monocytes and phagocytes move about a lot and get guided to the pathogens and squeeze through the tissue so that they can go there and do their jobs, which we're about to talk about soon enough. So once again, the types of white blood cells, you'll have the neutrophils, the eosinophils, and the basophils. Those, once again, are our granular cytes. And then you also have the monocytes and the lymphocytes. Well, the granular cytes and the monocytes respond to any threat, and they are part of our non-specific immune response. That's because any threat that's there, they're going to go after it. But the lymphocytes, they respond to specific threats. So they respond to specific pathogens and are responsible for a specific immune response. One of the chapters that we will be covering during this section is immunity. So we will be getting into how the nonspecific and the specific immune responses work. So let's start off with the neutrophils. These are the most mobile of the leukocytes along with the monocytes, and they make up about 50 to 70% of the circulating white blood cells. So they make up the majority of the white blood cells that are circulating. They are usually the very first white blood cells to arrive at the injury site, and that's because, again, they are so mobile. They are phagocytic, and that way they are able to ingest and digest bacteria. And these white blood cells, in particular these neutrophils, they increase in numbers whenever there's some sort of bacterial infection. So anytime you go and you get your white blood cell count taken, if they exceed the amounts, then that's when you have a concern that there's some sort of infection. If they exceed the typical count of 5,000 to 10,000. That's considered normal. So if that number changes, then you're going to have some sort of condition that is abnormal. The eosinophils, they only make up about 2.4% of the circulating white blood cells, and these are similar to neutrophils, and they are phagocytic, but probably weakly phagocytic is what I should say, but they are attracted to and kill certain parasites. They also control allergic reactions by removing biochemicals associated with the reactions. The basophils are a little smaller than the neutrophils and the eosinophils, and they are very rare as well. They only make up less than 1% of the circulating white blood cells. But the basophils migrate to damaged tissue and they release heparin, and the heparin inhibits clotting. They also release histamine, and histamine promotes inflammation. So this increases blood flow to the injured tissues. Now, the agranular leukocytes, the agranular sites, there are lymphocytes and monocytes. We're going to talk about the monocytes first. The monocytes are about twice the size of a red blood cell, and they kind of have a kidney bean type shaped nucleus to them. The monocytes, they usually make up about 2 to 8% of the leukocytes in a blood sample, and they live for several weeks or even months, and they are very aggressive phagocytes. They are macrophages that phagocytize bacteria, dead cells, and other debris, so they're very aggressive. The other type of agranular site would be the lymphocytes, and these are usually only slightly larger than red blood cells. A typical lymphocyte has a large round nucleus that's surrounded by some cytoplasm, a thin rim of cytoplasm, and these cells account for about 20 to 40% of the circulating white blood cells. And lymphocytes may live for years. The lymphocytes are very important to immunity, and some produce antibodies that attack foreign substances that are entering the body. The 5 white blood cells that we just covered, again, if you take the counts, the white blood cell count, they should fall within a certain range, and the range is typically from 5,000 to 10,000 cells in a microliter of human blood. If these numbers have some sort of change where they drop below the 5,000 or above the 10,000 count, I just realized I made it 1,000. Let me change that to 10,000 by adding a 0. If these numbers change, then that can signal some sort of abnormal condition. You can have leukopenia. That is when you have less than 5,000 cells per microliter of human blood. And diseases like measles, mumps, influenza, typhoid fever, chickenpox, AIDS, polio, those types of diseases will cause a reduction in the total white blood cells. You also have where you can exceed the 10,000 count, and that would be leukocytosis. And this is usually indicating an acute infection, such as appendicitis. And the white blood cell count, the total white blood cell count, is greatly elevated in leukemia. Usually a differential white blood cell count lists the percentages of the types of leukocytes in a blood sample. And that's useful because the relative proportions of white blood cells may change in particular diseases. For example, the number of neutrophils usually increases during bacterial infections, but the number of eosinophils may increase during certain parasitic infections and allergic reactions. In AIDS, the numbers of a certain type of lymphocyte drop sharply. So this can give you a very good indication of what type of infection is present. Okay, so soon we're going to be talking about in the next slide about the white blood cell formation. And I wanted to remind you of this figure that we looked at earlier when discussing the red blood cell formation. I mentioned that the hemocytoblast can develop into myeloid stem cells, and these myeloid stem cells can go on to mature. Some are destined to mature into red blood cells, but some of them are destined to mature into platelets. And then you have some of them that are destined to become granulocytes and also monocytes. Okay, so the hemocytoblasts can become myeloid stem cells, and these myeloid stem cells can become cells that will, the white blood cells, the granulocytes, and the monocytes. The hemocytoblasts can also become lymphoid stem cells, and the lymphoid stem cells can eventually become lymphoblasts and mature into the lymphocyte. So, based on what I just said, as a recap, you will have the hemocytoblasts, and the hemocytoblasts, those are regulated by colony stimulating factors called thymosins, and some of them can produce lymphoid stem cells. The lymphoid stem cells will differentiate into lymphocytes, and that process is called lymphopoiesis when making lymphocytes. And then these lymphocytes will migrate from the bone marrow into the lymphatic tissue. And some of them will produce antibodies that attack foreign substances that enter the body. The other white blood cells come from myeloid stem cells. The myeloid stem cells are the ones that can go into red blood cells, as mentioned earlier. They can mature into platelets, but they also become white blood cells as well. So the myeloid stem cells differentiate into all of the other formed elements, but of course right now we're talking about the other, the neutrophils, the eosinophils, the basophils, which are all granulocytes, and then they also become the monocytes. This slide simply gives you a review of the formed elements of the blood, starting with the red blood cells, and then we talked about the white blood cells, the neutrophils, eosinophils, and the basophils. All are granulocytes. And please feel free to stop right here and look at the abundance and the functions and the remarks on these, just to refamiliarize yourself. And then you have the monocytes and the lymphocytes, which are the agranulocytes. That means they do not have any granules in them, in the cytoplasm. And then the one that we have not quite spoken about yet would be the platelets. Those are the other formed elements within the blood. And the platelets are also known as thrombocytes. These are not complete cells, and we're going to talk about those next. So the platelets are not complete cells, and these platelets, let me write what they're also known as in case you hear them mentioned as such later on. They're also called thrombocytes, and these are not complete cells. They're little fragments of cells, and they come from large stem cells that fragment like a shattered plate, and they release small sections of the cytoplasm or of these, so we call them platelets because it's little, they shatter like a plate and they're like little plates, so we call them platelets, and they release these small sections into circulation, and they are involved in the prevention of blood loss. So you're going to see them involved in blood clotting. Shortly, we will go into the details of the clotting process, but the platelets do contain granules of chemicals that help initiate that clotting process. And the normal count for platelets would be between 150,000 to 500,000 per microliter of blood. If you have less than that, then that is considered thrombocytopenia, which is a low count. Okay, so this is the third time that I'm referencing this figure, and that's because now we are talking about how the platelets are made, and I just want to point out that they start off here. You have the hemocytoblasts, so that's again like that stem cell that's in the bone marrow that could differentiate into myeloid stem cells, and then the myeloid stem cells I know you heard it before, but one of the things that it can become would be this megakaryocyte. So the megakaryocytes are large stem cells that break up. These are the cells that break up. And here you can see that it's all shattered, broken up into little pieces. And this is what we call platelets. And the platelets is what go into circulation. The larger fragments of the megakaryocytes shrink and they become these platelets as they pass through the blood vessels in the lungs. So they are developed from hematopoietic stem cells. They lack a nucleus and they help close up breaks in damaged blood vessels. So, of course, while we are on platelets, this will be a great time to talk about hemostasis. Hemostasis is the stoppage of bleeding, and of course, you can well imagine that that is vitally important. When a blood vessel breaks, the platelets adhere to the collagen underlying the blood vessels. The platelets also adhere to each other and form a plug. A plug can control the blood loss in a small break, but a larger break will need a blood clot. Blood clots use a lot of biochemicals that we call clotting factors. So now we're going to talk about hemostasis, which is the stoppage of bleeding, and there are particular phases associated with hemostasis, and we're going to go through each one. The first stage of hemostasis is the vascular phase. The wall of the blood vessels have smooth muscle that are lined with simple squamous epithelium that we call endothelium. The endothelium, I hope you remember this type of jargon from our very first, when we went over histology, talking about simple squamous epithelium. When the wall of the vessel is cut, it causes a contraction. The contraction will cause the vessel to decrease in diameter. The contraction or the spasm slows the loss of blood, and this lasts for about 30 minutes. So when this decrease in the vessel diameter takes place, loss of blood is slowed. So I'll just write here, slows loss of blood. And this lasts about 30 minutes. The 30-minute period, this entire 30-minute period, is the vascular phase. The membranes of the endothelial cells at the injury site become sticky, and they stick together and may block the opening completely. The second phase is called the platelet phase. In this phase, the platelets begin to stick to endothelial surfaces and expose collagen fibers within 15 seconds of the injury. More platelets arrive and stick to each other, and this forms what we call a platelet plug. And this may be enough to close up a small break. The third phase is the coagulation phase, and this is what we refer to as blood clotting. The first two phases begin almost immediately. The coagulation phase does not begin until about 30 seconds into the event. Blood clotting results from a series of events leading from the conversion of fibrinogen to fibrin, and the fibrin is an insoluble protein. The blood clot effectively seals off the damaged portion of the blood vessel. So now we're going to talk about the entire clotting process and the cascade of events that we see taking place. And this cascade of events begins with the major event. The major event is the conversion of fibrinogen into the plasma protein called fibrin. The clotting process cannot occur unless the plasma contains the necessary clotting factors, which include calcium and 11 different proteins and vitamin K. Proteins are inactive and are converted to active enzymes that direct reactions in the clotting process. The clotting proteins interact in a sequence such that one protein is converted into an enzyme that activates the second protein. And because it is a step-by-step process, we call it a cascade. When a blood vessel is damaged, both the extrinsic and the intrinsic pathways are activated. Let's begin talking about the extrinsic pathway. The extrinsic pathway begins with the release of a lipoprotein called tissue factor. The greater the damage, the more tissue factor released, and the faster the clotting happens. So the extrinsic pathway begins with the damaged tissue releasing tissue factor. The tissue factor combines with calcium ions and other clotting proteins to form an enzyme which activates what we call the factor X. Factor X is a clotting protein that's made by the liver. The reason why this pathway is called the extrinsic pathway is because it is initiated with materials outside of or extrinsic to the blood. The material is the tissue factor, which is released by damaged tissue cells. So this is outside of the blood, so it's called the extrinsic pathway. The other type of pathway to blood clotting is the intrinsic pathway. And the intrinsic pathway is so named because it is initiated by the blood coming into contact with exposed collagen in the blood vessel wall. So we're talking about this pathway being initiated by materials that are within the blood or blood vessel wall. So it's called intrinsic. This is slower. This process is considerably slower, about 5 to 10 minutes, but it results in the formation of larger amounts of thrombin, and this allows the formation of larger blood clots. So the intrinsic pathway is initiated, once again, by the blood coming into contact with something that's inside of the blood or the blood vessel. In this case, it begins with the activations of proenzymes exposed to collagen fibers at the injury site. This pathway continues with the help of platelet factors that are released from the clumped up platelets. When we say aggregated, we mean that they are clumped up. Many other reactions happen within the intrinsic pathway that are also capable of activating the factor X. So in both the extrinsic and the intrinsic pathways, factor X is what is going to be activated, but it's just activated in different ways. With the intrinsic pathway, it is activated by the proenzymes that are exposed to collagen fibers. That's where it starts. So it's something that's within the blood or the blood vessels. And with the extrinsic pathway, the factor X is activated by damaged tissue. Either way, both of the intrinsic or extrinsic pathways will activate factor X, and that is the common pathway. Okay, factor X is the common pathway. So clotting is usually started by the shorter and faster extrinsic pathway or the intrinsic pathway, which is slower and reinforces the initial clot, making it larger and more effective. But regardless of which pathway starts the blood clotting, activates, it's all because of the activation of factor X. When factor X is activated, the enzyme prothrombinase forms. Prothrombinase is what converts prothrombin, which is a globulin plasma protein. It converts prothrombin into thrombin. Now remember, this is a cascade, so the product of one reaction is what's going to aid the other reaction. So here we have thrombin, and thrombin, in turn, thrombin will catalyze a reaction that joins fragments of fibrinogen into long threads of fibrin. And remember, the major event in blood clotting is the conversion of fibrinogen into the insoluble plasma protein fibrin. And I've written this out here already for us to follow along with to show you that fibrinogen is converted into fibrin, and that was done with the help of thrombin, and that thrombin was made because of the prothrombinase, which converted prothrombin into thrombin. This is going to stimulate, once fibrin is present, those fibers will stick to the damaged blood vessels, to the surfaces of the exposed damaged blood vessels, and creates a meshwork that entraps blood cells, and it also entraps platelets, and that resulting mass is a blood clot. And this is what is going to prevent blood loss. So just to recap this, you have the factor X, which forms the enzyme prothrombinase, and the prothrombinase converts the prothrombin into thrombin. Thrombin is what catalyzes the reaction of fibrinogen into fibrin, which thrombin stimulates formation of tissue factor and the release of platelet factor. The presence of these two further stimulates the extrinsic and intrinsic pathways, which is what we will call a positive feedback loop. In this figure, you can see the fibrin network forming, so fibrin threads have formed, and they stick to the exposed surface of the damaged blood vessels, and it creates a meshwork that starts trapping blood vessels, and you can also see platelets are trapped here as well. And this resulting mass is a blood clot, and this can block a vascular break and prevent blood loss. Sometimes after a blood clot has formed, there's a clear yellowish liquid that remains, and that's called serum. So serum is plasma minus all of the clotting factors. The amount of prothrombin in the blood is really dependent upon the degree of tissue damage. Once a blood clot begins to form, it promotes additional clotting. But of course, we do not want the blood clot to continue forming or to remain forever, because that can be dangerous as well. So the clot has to at some point be removed, retracted and removed. When the fibrin network is established, the platelets will contract. And when they contract, they're going to pull the torn edges of the wound closer together. So this is called clot retraction, and the whole point is to sort of pull the edges of the wound together. This makes the size of the damage smaller so that the repair is easier. As repairs continue, as they proceed, fibrinolysis takes place, and fibrinolysis is really just the breakdown of the clot. So this is where the clot dissolves. Damaged tissue releases thrombin, and it releases TPA. TPA is tissue plasminogen activator, and this activates plasminogen. Plasminogen produces plasmin, and plasmin is what digests a clot. It digests the fibrin strands and it breaks down the clot. With that said, this completes the section 4-1 lecture. I hope that you guys will read it over, look over it over and over again, read your books, look at your study guides, make notes, and really learn this information very well. If you have any questions, as usual, please make sure that you send me emails, and I will also go over highlights of this lecture during class. So please look at it before you come to class so that we can go over this lecture and you'll have an understanding of what you know and what you may not know. I'll see you in class. Take care of yourselves. Bye.